Okay, so first a minor disclaimer. So information presented is not the official opinion of Michigan State University or the University of New England. So I'm Zach Burton and my co-host Lei, hi Lei, are completely responsible for the content presented. So this is a Life on Earth, Origin of Life webcast. And I'm going to show you that type 1 transfer RNA evolution can be solved as a puzzle. So this is a 4 billion year old puzzle. And these are the presenters. Okay, so some subliminal messages. So type 1 tRNA evolution solved as a puzzle. It's a very old puzzle. It's about 4 billion years old, and it's etched in genetic code. It's a problem just about anybody can solve, so it's not a hard puzzle. It's actually a pretty straightforward puzzle, particularly when you have the result and solution in front of you. It's a problem in language, in evolution, in logic, and linguistics, at least RNA linguistics. I'm not sure I know very much about linguistics, but this is RNA linguistics. There are bottom-up and top-down approaches to origin of life. So a bottom-up approach would be making life in a test tube, for instance, and a top-down approach is inferring origin of life from existing biology. That's what we're doing here. We do it that way here because we're molecular biologists and we're interested in sequence and biology and molecular structures. Anyway, this is a problem just about anybody can solve, so it's not a very difficult puzzle. It's just a little bit hard to figure out how to approach it. I'm going to show you the 331 nucleotide mini-helix theorem. Theorem is a proven model, so I'm asserting that this is a theorem and not just a model, not just another idea or hypothesis, but in fact it's a proven theorem. And this is the solution to the puzzle. So you can use this as a cheat sheet to analyze the puzzle. This is recorded history from about 4 billion years ago. The history was recorded in genetic code and it was never overwritten. So there's a record of the original primordial tRNA sequences from pre-life before living systems. And it has to do with identification of root sequences. So we identify a primordial sequence for transfer RNA. So just to reiterate, there are bottom-up and top-down approaches to origin of life. So a bottom-up approach would be to generate life in a test tube. A top-down approach is inferring origin of life from existing biology, in this case, transfer RNA sequences. And it shows the importance of identifying root sequences. So we've identified a root sequence for transfer RNA. And we get at strange questions like these. So these aren't really scientific questions. Is there a God? Is a God necessary? But identifying a pre-life sequence for transfer RNA gets at such issues. Okay, in terms of tools, we're using Chimera X. That's a molecular graphics program. Transfer RNA database. And when you want to compare sequences, you want to align them. So you want to use a typewriter font like Courier. Chimera can be learned by running tutorials. It takes a few weeks to learn how to use Chimera. And the tRNA database is pretty straightforward and easy to use. So most tRNAs are type 1, and that's the type we'll talk about today. I could also talk about type 2, and we'll do that in a future webcast. It's a story of how life evolved on planet Earth, and it deals with chemical evolution of tRNA, with tRNA as a genetic adapter, and with polyglycine and protein synthesis. So I'm going to answer the question of where protein synthesis comes from. The root sequence of primordial tRNA is known with essential certainty. The sequence is completely ordered. That's 100% RNA repeats and inverted repeats. These are stem loop stems. The tRNA primordial sequence tells a compelling story about the origin of life. So transfer RNA is the genetic adapter. tRNA therefore links nucleotide sequence and protein sequence. Protein sequence is amino acid sequence, and the ratio is 3 nucleotides, a 3 nucleotide codon or anticodon in tRNA, and to 1 amino acid. Something to realize is that chaos does not evolve to order. So chaos evolving to order would be like monkeys typing Shakespeare. Okay, so that's not a good evolutionary model. Order evolves to order, and then order degenerates with time. And this is really the story of how tRNA pre goes to all the tRNAs of different organisms. tRNA, the primordial sequence, is 100% ordered. And that means it came from ordered precursors. We're going to assert that transfer RNA is the God molecule. So we're using God metaphorically here. God confers life on Earth, and tRNA confers life on Earth. The tRNA anticodon loop, we're going to call the God loop within the God molecule. The God molecule is tRNA. And presenting the 331 nucleotide mini-helix theorem. This is a theorem and not a model because it's a proven hypothesis. So type 1 tRNA. So this is an image of type 1 tRNA, and it's been colored according to the 331 nucleotide mini-helix model. And I'll just go through it so that you can familiarize yourself with the structure. So you have here a stem loop stem, stems of 5, loop of 7. This is the anticodon stem loop stem, and that's the wobble base, the middle base, and the third base of the anticodon. The anticodon 3 nucleotides in the anticodon refer to a single amino acid in a protein sequence, in this case, phenylalanine. So stem, loop, stem, and a homologous stem, loop, stem. So homologs are like identical twins, if you want to think about them that way. These are stems of 5. This is a loop of 7. That's a U. That's a U-turn. The U-turn is a U-shaped turn in the RNA backbone. Here you have a 7-mer loop, U-turn, U-shaped turn, and followed by the wobble base of the anticodon. So that's the wobble, that's the middle, and that's the third base of the anticodon. So this sequence here and this sequence here are still recognized as homologs, nearly identical twins, if you want to think about them that way. This yellow segment here, these 5, correspond to these 5 out of these 7. This is a 3' acceptor stem. This is a fragment of a 3' acceptor stem. And this is the variable loop of the transfer RNA. type 1 transfer RNA in this case, because it's a short variable loop of 5 nucleotides. And this sequence in green, these 5 correspond to these 5 here out of these 7 total. This is a fragment of a 5' acceptor stem. And this is a 5' acceptor stem here. And then this is the D loop. So I promised you that everything was repeats and inverted repeats, stem, loop, stem. So here's a stem, loop, stem, and homologous stem, loop, stem. This and this are related by genetics, just like twins or identical twins. These 5 here are derived in yellow, are derived from a CGC repeat. These 5 here, well, these 7 here are derived from a CGC repeat. In green, a GCG repeat. In green, derived from a GCG repeat. These 5 here. And this is derived from a UAGCC repeat, the D loop. Okay, these are called cloverleaf diagrams, and this is for an ancient organism, Pyrococcus. This is called a typical tRNA. It's like a consensus of all 46 Pyrococcus tRNAs. And this is tRNA glycine, encoding glycine. Glycine's the first amino acid encoded in the genetic code. And first I'll show you that this stem, loop, stem, and this stem, loop, stem are genetically related to each other. They're called homologs. You can think about them as twins or nearly identical twins. The sequence is CCGGG, CCGGG. 7-mer loop, U-turn after a U. So this is DNA sequence, so T in DNA is U and RNA. That's why I'm calling a T a U. In this loop here, that's a 7-mer U-turn after a U. So these 17 here, 5 plus 5 is 10 plus 7, 17. The same thing here, 17. These sequence and this sequence are very similar. And they're very similar after 4 billion years of evolution, separate evolution in 2 different parts of a molecule on Earth. So that's really remarkable conservation of these 2 sequences. This sequence here, this interval, is also 17, and it's based on a U, A, G, C, C, U, A, G, C, C, U, G for A, G, C, C, U, A, U, A, G, C, C repeat. And this sequence here, G, C, G, G, C, G, G, a G, C, G repeat. It's CGC complement. This used to be GGCGG. It's only slightly changed, as in GGCGG there. This sequence here used to be CCGCC, as in CCGC, often C. tRNA glycine is the first tRNA. This sequence is almost identical to this sequence. And what that means is that primordial tRNA was a tRNA glycine, and all the tRNAs of an ancient organism are derived from a primitive tRNA glycine. Just to show you that one more time, this is the primordial tRNA sequence. This is the consensus sequence called a typical sequence. It's a summation of 46 tRNAs in Pyrococcus, and this is glycine tRNA. So primordial tRNA is a primitive glycine tRNA, and all tRNAs in one of these ancient organisms is derived from a primordial tRNA. So just to reiterate, so this is a GCG repeat, GCG, GCG, G, and this is a GCG repeat here, this little green segment corresponding to those 5. This is a UAGCC repeat in the D loop. This is a stem loop stem, and this is a stem loop stem. You have a U-turn after a U here. So B indicates G, C, or U, never A. There's never A in a wobble position. That becomes important at some point in these discussions. And let's see, what else am I thinking of here? Stem, loop, stem, homologous, stem, loop, stem, U-turn after a U, 7-mer loop, 7-mer loop. And these sequences that you see here are essentially the sequences from the last universal common ancestor, affectionately known as LUCA. And the variable loop, that's a CCGCC corresponding to CCGCC. This is a CGC repeat, 7-mer, GCG repeat. This complements that, and they form the acceptor stems of the tRNA. So this is the solution to the puzzle. This here, stem, loop, stem, stem, loop, stem, stem, loop, stem, stem, loop, stem. This is a CGC repeat. This is a GCG repeat. This is based on a CGC repeat, GCG repeat, UAGCC repeat. This is what's called the discriminator base. That's an A initially, and then it diverges to all different bases. And then you have a 3' A, you have 2 Cs to hold the tRNA on the ribosome for protein synthesis. They form Watson-Crick pairs with Gs. And then here you get phenylalanine attached to the ribose ring here of the 3' A. So these 3 bases are consistent tRNA to tRNA. The discriminator base can change. And this is just more of the same. So here you see this stem loop stem. I'm arguing that it's similar to this stem loop stem in this organism in a typical tRNA from Pyrobaculum. The sequence is CCGGG, CCGGG, so a perfect match. Here it's not quite perfect. And here you see CCGCC, as in CCGCC. This is slightly changed in evolution. And this was GGCGG, GGCGG, slightly changed. And the reason this sequence is slightly changed is so it can pair here in the D stem to form the cloverleaf fold. And this is supposed to be an A, but it's not an A because it intercalates between these 2 A's and forms a hydrogen bond to a U over here. So that's why that doesn't fit the model. That's really one of the only systematic differences between the model and sequences in living organisms. And here's a bunch of statistics that says what I just said. So every feature of the theorem is correct and demonstrated by statistical tests without details. All of this is published if people are interested. Okay, so this is the 331 nucleotide mini-helix theorem. So why is it called that? So here are 3 different mini-helices, and tRNA was made out of these 3 mini-helices. So each of them is 31 nucleotides. They have 5' acceptor stem, which is a 7-mer, and a 3' acceptor stem, which is a 7-mer. And here you have a 17-mer. This is a UAGCC repeat that becomes the D loop of tRNA. And this stem loop stem and this stem loop stem are initially identical. There's some ambiguity in the red segments about exactly what the primordial sequence was. If you take this and you ligate it to that and you ligate it to that, you get this. That's 93. That's 3 times 31. This becomes the D loop, anticodon loop, and T loop. So you string these all together like that. If you process here, here, and there, you get more mini-helices. So this is a mechanism to replicate mini-helices. And if you process here by a 9-nucleotide internal deletion, so you remove these 9 nucleotides, you're left with GGC, GG, GGC, GG, GGC, GG, GGC, GG. So that's how you process to this sequence. It's the same in type 2 and type 1 tRNAs. And no processing in type 2 tRNAs. So this is a 3' acceptor stem ligated to a 5' acceptor stem. And if you delete these 9 nucleotides there, those 9 nucleotides there, you end up with CCG, CC, as in CCG, CC, CCG, CC, part of a CGC repeat. And there are 4 points to make about this system. The first point is that it's a 100% ordered sequence. That is to say 100% repeats and inverted repeats, unless you're considering ACCA, which seems to be a primitive adapter to attach glycine. Second point, the anticodon 17 nucleotide stem loop stem and the T 17 nucleotide stem loop stem are homologs. So what did I just say? So I just said that this stem loop stem and that stem loop stem, these are initially identical sequences. They're identical here. By the time you get to tRNA, they change slightly, but only in the loops. So this is a twin of that. And then the third point is that there are 3 mini-helices, so you can count them various ways. So 1, 2, 3, 31 nucleotide mini-helices, or you can count them 1, 2, 3, according to the mini-helix cores. And then the fourth point is that I've completely described type 2 and type 1 primordial tRNA sequences. So there are competing models for tRNA evolution, but I would avoid them if I were you, and none of them will rationalize the differences between type 2 and type 1 tRNAs. And the other thing to realize about this is all chemical evolution. So we think of Darwinian evolution with organisms eating organisms or organisms outcompeting other organisms. But everything on this page is older than Darwinian evolution. It's older than life. It's all pre-life, and it's all chemical evolution. And so, because this is all chemical evolution, what drives it? Because it's not organisms eating other organisms. So what drives it, at least a big part of what drives it, is to synthesize polyglycine. So mini-helix world pretty clearly came from polymer world, where you have the same repeats and inverted repeats. And God knows what else. Okay, you had other things too. But this is a primitive system to synthesize polyglycine. And mini-helix world is a improved method to synthesize polyglycine. So that's a driving force to go from polymer world to mini-helix world. And then the driving force to go to tRNA is this is a better means initially to make polyglycine. But this is a system that can learn to encode proteins. And through evolution, it learns to encode proteins of whatever sequence. So we've spoken about the God loop within the God molecule. Okay, so we're using God metaphorically here as the force that creates life on Earth. And the God loop is the anticodon loop. The anticodon loop is particularly important. It's a 7 nucleotide loop with a U-turn separating loop positions 2 and 3. It's the most important biological intellectual property on Earth for evolution of life. So if you're trying to create life on some moon or planet somewhere out in a galaxy far, far away, you pretty much, if you're going to do it out of RNA, you pretty much need the 7 nucleotide loop. There may not be any other way to do it. The genetic code evolved around the tRNA anticodon, and this is really an important structure. And so it's important to look at and to at least in part understand. And so here it is. So these 7 are the anticodon loop. And here you have an A and a C. The C is methylated, but that's because this is a tRNA from brewer's yeast in this case. So anyway, you have a Hugstein hydrogen bond here. That's a weak interaction. You have a U, you have a U-turn, you have a wobble base, you have a middle base of the anticodon and a third base of the anticodon. Then you have a highly modified base here. And because this base is highly modified, it can't be part of the anticodon. So the anticodon can't be 4 bases, has to be 3 bases. So this helps to keep the frame for translation. But in addition to that, this highly modified base is part of the reading of this 36 base. So you have 34, that's the wobble position, 35, that's the middle position, and 36. And these 3 bases relate to the structure of the genetic code. So in a future webcast, we'll discuss the genetic code in more detail, but here I'll just partly introduce it. So this is a genetic code with a complexity of 32. So in textbooks and in courses, you'll learn that the complexity of the genetic code is 64, but it's really 32. It's 32 because at a wobble position, you only have purine versus pyrimidine resolution. So basically, U and C are too close to each other to be separated into 2 amino acids, and A and G are not really separable in terms of the wobble position. So there are only 2 possibilities here, purine versus pyrimidine. For here, that's a Watson-Crick position, AGCU, and that's a Watson-Crick position, AGCU. So 2 times 4 times 4 is 32. This is a complexity 32. This is the codon here, first position. second position, third position, and purines and pyrimidines are not discriminated in forming the code. And this position here refers to columns 1, 2, 3, and 4. There's lots of evolution in columns. Just to give you an idea of this, these are a bunch of hydrophobic amino acids, and they're all 1A aminoacyl tRNA synthetases. Those are the charging enzymes for tRNAs. This is the anticodon, and U is considered to be essentially equivalent to C, and you never use A, never A in a wobble position. And G works because G can pair with U as a wobble base, U, or with C as a Watson-Crick base. So you don't need A, you can use just G, but this is the source of degeneracy in the genetic code, because there's wobble ambiguity, there's degeneracy in the genetic code. These in red, serine and threonine are related amino acids. These are all 2A amino acids, 2A aminoacyl tRNA synthetases. Here you get this lovely stripe pattern, orange, green, orange, green, orange, green. That can be explained. And here, arginine and cysteine use 1A enzymes that are closely related. So all that can be explained. The wobble position relates to the A and B rows. So here you're getting sectoring along A and B rows. So you're getting sectoring according to the middle base and the wobble base. And here, rows 1, 2, 3, and 4, that relates to C. This is the 36th position, that's 36, C, U, G, A. And row 1 is late to fill because initially this was a wobble position and then it wasn't allowed. When this is a wobble, you can't have A here, so you can't fill row 1. So row 1 fills late in genetic code evolution. So I hope I'm getting across the idea that these three bases relate to this table in a complex way. Takes a little getting used to fully feel comfortable with this. But genetic code evolved around the anticodon. That's the tRNA anticodon. And degeneracy at this position, and initially degeneracy at this position, gave you the structure of the code that we have today. Lots of evolution around the middle base. You see that here with all these yellow amino acids and green and red amino acids and this stripe pattern. And here a couple of cyan amino acids. Got all that? Me neither. Anyway, this is a discussion for another day, but I wanted to introduce it here to emphasize the importance of these 7 nucleotides. If you don't have those 7 nucleotides, you don't ever get a genetic code on Earth. So that's important. So what I think I've told you is that polymers, regular polymers, repeats and inverted repeats, go to regular minihelices, repeats and inverted repeats, stem loop stems, and then you go to primordial tRNA, type 1 and type 2, and from that you go to tRNAomes, all the tRNAs of an organism. From that, genetic code, aminoacyl tRNA synthetase enzymes, proteomes, and eventually to life. But tRNAomes, the genetic code, aminoacyl tRNA synthetases, and proteomes, at least primitive proteomes, are all co-evolved before you get life. So we're talking about pre-life here. We're not really talking about living organisms. We're talking about chemical evolution. So from my point of view, life on Earth and origin of life is largely a solved problem, at least at the level of outline. lots of details missing, but the outline is really very strong and it penetrates deeply into pre-life. And if you're interested, I have a Substack that deals with some of these issues. I explain how life evolved on Earth, and it's a good puzzle. It's free. And that brings us back to our title page and our subliminal messages. So anyway, I'm hoping that all this is of interest. Some people don't like biology, but they like language and logic and maybe even linguistics, whatever that is. So anyway, that's it. That's all I have to say.
Hey, Zach.
I like
So,
how we
yeah,
doing?
there are several questions I want to ask you. Some of your audience may have similar question in mind as well. Okay. My first question, can you discuss RNA as a language?
Okay, so RNA is kind of a simple language with A, G, C, and U as bases. It turns out that co-evolved with the genetic code or tRNA modifications, so it becomes a little bit more complicated than that. But really, to solve this particular problem, all you need is A, G, C, and U. And it's a simple language and it's a simple result. Repeats and inverted repeats. That's all you need to build tRNA.
So to follow on that, because RNA is a written language, if some sequence have not been overwritten, there's a recorded history of pre-life events.
Right. So the whole idea behind the 331 nucleotide mini-helix theorem is that it's 100% ordered repeats and inverted repeats, except for the ACCA adapter at the end. So um, basically, that's a written history. It's recorded that tRNA was generated from 331 nucleotide mini-helices, and two of those were initially identical to become the T loop and the anticodon loop. The D loop was a different flavor, but a related molecule.
My other question is, can other pre-life sequence record besides tRNA be recovered?
Yeah, so what I would say about that is that so far I don't know of any. Um, tRNA doesn't change very much, at least in the most ancient organisms, the ones that are most similar to the last universal common cellular ancestor, LUCA. And tRNA doesn't change very much because it forms the functional core of the translation system. So basically, its evolution tended to freeze early in evolution, and things changed around it, which is kind of an interesting result. So we've tried to do similar things to what we did with tRNA, for instance, with ribosome sequences, and sort of failed spectacularly. I, I don't know that it can't be done, but so far we haven't been able to do it. So there may be other examples, but I don't know what they are at the moment. tRNA is the only thing I can think of that appears to be unchanged. And we can tell it's unchanged because it's still so highly ordered because of the repeats and inverted repeats. If tRNA wasn't made of repeats and inverted repeats, I wouldn't be able to tell the story.
So in your presentation, you mentioned several tools, including Camera X. Actually, can you describe a little bit more how to use Camera X to analyze tRNA structure?
Okay, so that's kind of a complicated question, and I don't want to go into it in too much detail here. But I do want to stress that molecular graphics is wonderful for learning about chemistry, tRNA, protein structure and function, just about anything that I would consider interesting, you can address by using Chimera X. So it's very worthwhile to spend a couple weeks working tutorials and learning how to use the program. Just taken as a computer game, it's the best computer game on earth. So I'm a big fan of Chimera X. And it doesn't take that long to be as good at it as I am, because I'm no expert. I'm on level 0 of this computer game, and I may never progress from there.
I have another question. So, why is a genetic adapter important for evolution of life?
Okay, so um, I have no idea if there's another way to generate living organisms, but I don't think that there is. I think to generate living organisms, you need a molecular adapter. And the only molecular adapter that I can understand at this point would be transfer RNA. And it has to be transfer RNA, or it has to be very close. So if you're sampling the nightlife on Enceladus and you meet an alien, you should find out what the sequence of their genetic adapter is. So it's either like tRNA, and you can think about panspermia, or if it's different than tRNA, I can't imagine what it is. If it's made of RNA, it almost has to be similar to tRNA because it needs that 7 nucleotide loop. If you don't have the 7 nucleotide loop, you can't get a 3 nucleotide genetic code. And you can't have a 4 nucleotide genetic code because you could never evolve it. And you can't have a 2 nucleotide genetic code, so far as I know. I mean, it might be conceivably possible, but it poses a lot of problems. So it's very difficult to think of another way of generating organisms with proteins that are as diverse as the proteins that we associate with life. So to get chemical complexity in life, you need a 3 nucleotide code based on 4 nucleotides. I don't think there's another way to do it.
Another question, when you say life on Earth is a largely solved problem, what do you mean by that?
Well, I would say it's mostly solved in outline. So, and the outline is really so good. I mean, it's a surprise that the outline is as good as it is. I think what I've presented in this talk is really impactful and important science. That basically, if you think of polymer world and mini helix world and tRNA world, these must have been very complicated worlds with lots of polymers and lots of molecules and lots of small molecules and metabolism of various sorts. But when you take a top-down approach, you're picking the winning strategy. So transfer RNA and mini helices and polymers, that turned out to be the winning strategy. I don't think we completely understand why yet, but have lots of ideas. But you could certainly go from the information in this talk and do a better job of making life in a test tube. That's one thing I would say. I have lots of suggestions for the bottom-up people from our work on tRNA sequence.
Finally, how is chemical evolution in pre-life different from Darwinian selection of living organisms?
Well, that's another good question, and I don't really know the answer to it, but so chemical evolution relies on something that I refer to as pseudosymmetry. That is, you take similar molecules and you connect them if they're RNAs, and connecting those RNAs together, then you can fold them into more complex RNAs like transfer RNAs. And if you make polyglycine. Polyglycine is clearly important in the pre-life world. I don't know all the uses of polyglycine, but I think what it did was make better protocells. But basically, there's there's a very strong driving force behind chemical evolution. Otherwise, you would never have gotten life on this planet. And it's pretty fascinating, and it could be studied in a laboratory. Not my laboratory, because my laboratory is closed, but somebody else's laboratory could pick this up and run with it. There are lots of good projects here.
Thank you very much, Zach.
Well, thanks, Lei. And thank you all for listening. And we'll sign off.